this on. I was a budding chaparral scientist at the last symposium, just about to start my PhD, and in the time it took for this next one, I managed to finish. And one of my goals at that, seeing all the great talks, was I'll know I'll made it in the chaparral community when I'm invited to give a talk. And so now, after my short tenure as a doctor, I can retire. But I'm going to show you all today some of the work from my dissertation. Maybe. There we go. So first I want to thank everyone who's helped out on the projects, my multiple funding sources. And then the work I'm going to show today took place in the same area of Lake Piru in Ventura County on the southeastern edge of the Los Padres. And it's really addressing these two components of the complex problem that Carla identified. And so when we work in steep habitats, we know that it's a lot harder to do consistent watering. We can't get machinery or equipment up there. So as you'll see, any watering that we do, we have to hike up. And then the habitat is heterogeneous. And so that begs the question of if there's similar survival across the whole landscape, or there's these microsite conditions that are actually driving survival. And so the work I'm going to show you today actually didn't take place on that slope. It took place on a slope that, believe it or not, was steeper. And we can visualize this slope in 3 dimensions. And we can start to see how it's heterogeneous and how that heterogeneity in ridges, valleys, different elevations, different facing slopes actually drive the greenness of the annual grasses here in the springtime, where we see areas that maybe hint at more arid sites where the grass is already senesced, it's green, or it's brown, or more mesic sites where the grass is still alive and still green during this late spring period. And so this begs the question of, in a restoration context, where and why do different species survive, specifically at the microsite level? And so for today, I'm going to focus on 2 main aspects of the microsite: environmental conditions. And so we can imagine if we have a plant like this rooted in the ground, it's going to experience stress at the microsite level below ground with this more traditional ecological drought we think of in soil moisture. But it also is going to experience stress above ground, the leaf level, with this variable of vapor pressure deficit that's gotten more recent attention as this atmospheric drought. And for those of you who aren't as familiar with what vapor pressure deficit is, it's this integrated measurement of both temperature and relative humidity. And so we know at a given temperature, the air, represented by this blue balloon here, can hold a certain amount of moisture before it becomes saturated. However, as air heats up and expands, the same amount of moisture is no longer saturating the air. And that difference in the amount of moisture in the air versus the amount it takes to saturate it at a given temperature is the vapor pressure deficit, with larger deficits being more stressful at the leaf level. And so now that we know how we're characterizing these microsites, we can get to our question of how do microsites affect restoration survival, with the main hypothesis that microclimate and plant traits are going to interact to determine species performance, quantified both through survival and growth, but for today, I'm only going to talk about the survival. And we measured a lot of traits like Carla showed. I'm going to focus on 2 main traits today. First, this commonly referenced suite of traits known as the leaf economic spectrum that ranges from more conservative species like our chamise with a lower specific leaf area, and so you can see these are small, thick leaves, lower leaf nitrogen per unit mass, with leaf nitrogen generally being thought of as photosynthetic material, and so lower photosynthetic capacity, but longer lifespans. And so the leaves on chamise might last a year, 2 years, last for a longer time on the plant. And then that spectrum varies all the way to the more acquisitive side, like our brittle bush, with a higher specific leaf area, so you can see it's much larger and thinner, higher leaf nitrogen per unit mass, so more photosynthetic capacity, but shorter leaf lifespans. Species like this are actually drought deciduous, and so they generally only have leaves on for 6 to 8 months, depending on the season. And then the other trait that we're going to look at today is delta 13C water use efficiency. It's thought of as an integrated view into the water use efficiency of the material for its lifespan. And we can think of this as essentially carbon in versus water out, where if you have a plant like our elderberry with lower water use efficiency, it's going to have its stomata open to a certain degree, and that's going to allow it to assimilate some amount of carbon through photosynthesis, but as a result, it loses some amount of water out through transpiration. Whereas a species like our sugar bush with a higher water use efficiency might have a more closed stomata, be able to assimilate similar amounts of carbon in, but as a result, lose less water through transpiration. And so those are the traits we looked at, and we did this study on a slope shown here with some of the site visible from the road. Again, you can see that habitat heterogeneity and how, again, that's driving different colors of the annual grasses, hinting at the differences in aridity throughout the site. And at this, we planted a little over 1,600 individuals of 10 different species. And for each planting location, we weeded out a 1/2 by 1/2 meter square and then staked down this weed cloth for more long-term non-native exclusion so we could get rid of the biotic competition and isolate these abiotic effects that are determining survival and growth. Then we got to hike all of our plants up, 40 plants at a time, put them in the ground. Here's some of our plants right after going in the ground. And then a note on the joy of watering. Like Carla said, we didn't water intensively, but we did water at time of planting, and then twice more during the growing season because of a month rain-free period. And so we got to hike up all of that water, 5 to 7 gallons at a time. And while we did try installing rain catchments to reduce the amount we had to hike up, because we were watering due to a rain-free period, they were inherently not that successful. Here's a few more shots of plants right before going into the ground. So you can see they're quite robust, both above and below ground, with some root systems extending beyond 20 centimeters. You might even be able to see a little lignotuber forming on that russovato over there. And so these were developed plants. These weren't tiny little young plants going in the ground. And then in addition to tracking survival and growth throughout the study period, we also GPS to sub-meter resolution all of our 1,600 planting locations. So we could append a variety of 1 meter resolution topographic variables. And then we measured hourly temperature and hourly humidity at a subset of locations and daily soil moisture. And this allowed us to then use this microsite environmental variable data and our topographic data to create predictive models to look at environmental conditions of soil moisture and VPD over our whole landscape. And so we can now look at whether or not our hypothesis held true. And so first, looking at general survival of just all of our individuals together, with survival probability from our survival model on the y-axis and time on the x-axis, we see a general trend of die-off starting the end of May, and so the beginning of this hot, dry season, and then ending around mid to late November with the onset of first rains. And indeed, if we zoom in on this and then look at nearby weather station data during this time period, we can see that it coincides with a dramatic uptick in daily maximum temperatures and the cessation of any rain. And so indeed, this hot, dry summer characteristic of the Mediterranean climate we're working in is driving first-year mortality. But what does this look like when we get into individual species? And so next, I'm going to show you a graph where we're looking at this mortality index on the x-axis for each of our species, with our highest surviving species, Chaparral yucca, as the reference species represented by this dashed line at 1. And so values greater than 1 that don't overlap with 1 indicate that species had higher mortality than our reference. Values that overlap with 1 indicates they had no difference in mortality. And then while I won't see this because I picked the highest surviving species as our reference for completion's sake, values less than 1 would indicate lower mortality. Furthermore, for all of our species, non-overlapping confidence intervals also represents significant differences in survival, which are going to be referenced by significance letters. And that's colored by species by the 2 vegetation communities they're most commonly associated with. And the main takeaway here is when you actually look at individual species, there isn't a strong separation of sage scrub and chaparral. Some of our chaparral species survive just as well as our sage scrub species. One other note I want to make is laurel sumac has a lot lower survival, and I think that's because this is one of the few winters where we actually had below freezing temperatures in the winter. And Brandon Pratt and Anna Jacobson have shown that laurel sumac is especially vulnerable to these below freezing events. And so if we can't necessarily just group our species by sage scrub and chaparral and overall predict how they're going to do, this is where the role of plant traits might come into play. And so first, looking at modeled results from a predictive model that I made from all of our data, looking at the range of VPD across our site, mean centered on the x-axis and survival probability on the y-axis, with probability ranging from less likely to live to more likely to live, and then less stressful to more stressful for VPD. And for this modeling, we held soil moisture constant at a moderate value. And then we're going to look at 3 representative species here, ranging from our more water-efficient sugarbush to our less water-efficient elderberry. And so if we look at our less water-efficient species first, we can see that at the most stressful VPDs, it has virtually 0 survival. A similar trend for our intermediate species, but our more water-use-efficient species has significantly higher survival at even the most stressful VPDs that we measured. Similarly, if we now do a similar exercise for soil moisture, holding VPD constant at a moderate value, ranging from drier to wetter, and again, mean centered, and look at 3 characteristic species of our leaf economic spectrum, ranging from our more conservative chamise to our more acquisitive brittle-bush, we see that our more conservative species has low survival at our driest microsites, a similar trend for our intermediate species, and significantly higher survival at the driest microsites for our most acquisitive species. But we also know that traits don't occur in isolation, and in fact, for our species, there seems to be this trait trade-off, or trade-off if you will, where our most acquisitive species also generally had our lowest water use efficiencies. And so what that means is for our high water use efficient species that are unaffected by VPD, because of that trade-off, they're inherently going to have lower survival at drier soil moistures. And similarly, while our most acquisitive species can survive those drier soil moistures, because of that trade-off, they're inherently going to have lower survival at our more stressful vapor pressure deficits. Lastly, if we look at these 2 graphs together, we can see that the significantly lower survival of even our best species at VPD might indicate that VPD is a stronger driver of mortality than soil moisture, at least at our site. And so kind of summarizing, we see that there's this interaction between water use efficiency and vapor pressure deficit, and the leaf economic spectrum and soil moisture, but like I just showed, VPD might be a stronger driver than mortality. And so we're seeing how, well, if we look at all of our survival across all of our landscape or multiple landscapes, we might identify a better suite like sage scrub, but that's not necessarily the case when we get down to the microsite level. And so we can actually start to think about how we can break up the environment through areas that are going to have both low VPD and high soil moisture, and most of our species are probably going to survive there. Whereas sites that have high VPD but moderate soil moisture, our more water use efficient species will be able to survive there. And similarly, areas with moderate VPD and low soil moisture, we can get our acquisitive species. But I don't want to end on a sad note of what about this area. I want to offer a little glimmer of hope. And so I'm going to very briefly show a result from another study that Carla hinted at, looking at how nucleation or planting species in higher density might impact survival. And so we find that if we look at our initial planting density, we have significantly higher survival of our sage scrub species as we increase planting density, and no real strong effect of our chaparral species. Furthermore, when we look at the effects of traits, it looks like that this higher planting density is really ameliorating VPD at the microsite level through a similar effect that's been documented in forests, where the canopy can have shading, allow this kind of more hanging of fog or relative humidity. And so that we think that's why here it's really benefiting these sage scrub species that are generally more acquisitive, and as we saw, are more affected by VPD. And so we have a glimmer of hope hidden in the shrubs that we might actually not only use traits to predict where we can get plants to survive on the landscape, but then use traits to inform new strategies that will expand where they survive. Thank you.